Well, good morning. I'd like to thank the organizers for inviting me and also compliment them really on the order of presentation. So yesterday afternoon, if you were here, you were fortunate to see a really nice talk about how PD-1 and some other checkpoint molecules work using human cells in vitro, and that was Dr. Zaror showed some really beautiful data. What I'm going to do this morning is I'm going to talk to you about how PD-1 and related agents work on mouse cells, mostly mouse cells, in vivo models of tolerance, and a little bit get at the mechanism of how this works. And then what will happen next is Dr. Topalian, one of the folks I actually most admire in the world, frankly, will take it home and she'll tell you about how this works in human cells, in humans, in vivo. Okay? So we'll start out. These are my, these are my required conflicts of interest, theoretically. So I'll start out by talking a little bit about the similarities and differences between CTLA-4, which most of you are well acquainted, and with PD-1. But then I'm going to start to talk about how PD-1 is upregulated and how it is a marker for chronic antigen exposure. Chronic antigen exposure, of course, in most models leads to exhaustion. So by the transitive property, PD-1 is a marker of exhaustion in many systems, but not always, actually, and I'll show you how and why. I'll talk about how blocking PD-1 works immunologically in terms of cell numbers, but I'm going to introduce you to a concept which maybe you haven't thought of before, and that is that maybe PD-1 works a little bit differently. Maybe PD-1 works in the priming phase of T-cell activation. And then finally, I'll talk a little bit about how LAG3 and PD-1 interact both in mice and in other models. So this is a lovely slide. It's by somebody you might have heard of, Dr. Rebus, actually, showing potentially the differences between CTLA-4 and PD-1. And the idea, actually, the simple idea, is that CTLA-4 operates during the priming phase of T-cell activation. That is, when a T-cell is activated, it needs two signals, signal 1 through the T-cell receptor and signal 2 through CD28 from B7 molecules. When the T-cell upregulates, and the exact mechanisms that lead to upregulation of CTLA-4 are not completely clear, frankly, but in tumors and in tumor-draining lymph nodes, it's upregulated. When that's upregulated, it hijacks. It binds to B7 molecules with higher affinity than CD28 does. So essentially hijacks signal 2. If you block that with a monoclonal antibody like ipilimumab or tremelimumab, you allow signal 2 to proceed, and you can wind up with T-cell activation. PD-1, on the other hand, might or probably was thought to act more in the effector phase. That is, when a T-cell sees its target that it's going to lyse by in either tumor or perhaps in a viral model, that PD-L1 is upregulated on some of these tissues and it protects them from lysis from PD-1 positive cells. And if you block this, you can allow effector function and lysis to continue. Now recently, actually, Alan Corman's group published first, and then Jim Allison followed up, showing that maybe CTLA-4 is a little more complicated than we thought. That maybe CTLA-4 antibodies deplete regulatory T cells, and maybe this is an important part of their mechanism of action. I'm going to show you something a little similar too, actually. I'm going to show you some data suggesting that maybe PD-1 operates also in that priming phase, and maybe that's an important part of its function. So again, just the idea that things aren't always as simple as you think. The reason that PD-1 is interesting is because the clinical effects are really quite astounding sometimes. And this is, when it's a personal patient of yours, it always feels different, right? So this is a patient of mine, a 57-year-old gentleman with advanced kidney cancer. You do not need to be a radiologist to see this advanced kidney cancer, which was refractory to all the targeted agents that we like in kidney cancer. And he had been through them all, and he's actually giving up, frankly. He was done. He was not interested in any more treatment. He had heard of something. He came to Hopkins, and he wound up enrolling in a phase 1b trial of anti-PD-1. After 6 months, the tumor had shrunk quite reasonably. He was less fatigued. He was enjoying. He actually went back to work. He was a teacher. And then it was interesting is this trial specified, and Suzanne will tell you a lot more about this, specified treatment for 2 full years. He tolerated 2 full years of anti-PD-1 every 2 weeks with really no major side effects. And at the end, there were still these small residual masses. So we were really curious about what was there. We went on and did a PET CT scan, and it was completely negative, completely negative. So he has now he has a PET complete response. He's about 6 months off of treatment, actually, and we continue to follow him, and he continues to do well. So this is why we're interested in PD-1 and both how it works mechanistically in mice and also clinically in humans. So Dr. Zaror actually mentioned this many, many times, but I just wanted to show this so that everyone gets the feeling. When a T cell divides, it upregulates PD-1, okay? And these are, this is CFSE labeling. These are undivided cells, and these are two different ways to activate T cells in vivo. And you can see in this way, PD-1 comes up a little bit later than this way, and this way it comes up and it continues to go up. What can you tell me by looking at that about those function of those T cells? Is one exhausted and one not? Is one activated more than another? What is the eventual fate? The answer is you really don't know. It turns out that these cells that have a lot of PD-1 are not necessarily more activated. These have less interferon gamma production. These cells actually have a higher interferon gamma production. So again, if you just see PD-1 on its cell without looking at other indices of effector function, you do not know whether it's a cell that's already exhausted, a cell that's going to be exhausted, or a cell that's been properly primed. And in fact, this is what's happening actually. So this is a model of self-antigen or tumor antigen recognition where T cells upregulate PD-1 and it remains elevated because of chronic antigen exposure. In this model, this is a vaccine. This is Vaccinia vaccine. We see this with other vaccines. And what happens is PD-1 goes up and then goes down, actually, mostly down. A population of memory cells will keep it up. But the bottom line is just by looking at PD-1, that does not tell you whether it's an exhausted cell or a recently activated cell or how activated that cell is. If you look in tumors, actually, and this is true, Dr. Zaror showed this nicely for melanoma, the T cells that infiltrate tumors, the CD8s, this is from prostate cancer, are mostly PD-1 positive. He showed 95% in some of his cases, I believe, actually, and this is probably universal finding in many tumors. I think what's a little less appreciated is that if you look in the peripheral blood of cancer patients, PD-1 is also upregulated, actually. So these are prostate cancer patients, and patients with more advanced disease tend to have more expression on their cells. This has also been investigated in kidney cancer, and there'll be an important publication out this from Kerry Campbell in the next couple of months, I believe. So again, it's up not only in the T cells in the tumor, but also in the peripheral blood. And again, it's a marker of antigen exposure just by seeing it. And actually, Rafi Ahmed published a beautiful paper on this where he took T cells out of the peripheral blood and looked at the PD-1 positive cells versus the PD-1 negative cells. And really, just by looking at PD-1, it doesn't prove the cell is exhausted. So I hope I'm making that point because I keep saying it over and over. So what does it do? What does blocking or lacking PD-1 do? And you saw clinically, right? And it was really pretty spectacular. You know, in vitro and in vivo, it basically, when you block PD-1 or you lack PD-1 genetically, you wind up with more cells with more effector function. But it's not as spectacular as that patient would show you. So this is a model where we adoptively transfer specific T cells. We see the same answer with T cells specific for several antigens, including GP100 or hemagglutinin or OVA. So you put in the cells, you vaccinate, and then you see what happens to basically cell number or cell function. We measure cell function using a CTL assay, an in vivo CTL assay. So bottom line is if you put in these cells to a self-antigen model, and better than using antibody, we have knockout cells, you see a fair increase in the number of cells, and they're reasonably more functional. And that's good, actually, and that's what Dr. Zaror actually showed in vitro using human cells. In tumor antigen models, these are mice that express an endogenous tumor antigen. These are mice that have hemagglutinin in their prostate. We put in prostate-specific CD8 cells. We prime them. We put in targets. And they really, honestly, cannot kill at all. Cannot, okay? In the presence of the tumor in these mice, these are not even large tumors. These are mice with 12 to 14-week-old tumors. The T cells cannot kill, but if they lack PD-1 or you block PD-1, they now acquire effector function. So a reasonable increase in cell number, but perhaps a more notable increase in the cell effector function. So how does this work, actually? And again, the easy answer is Dr. Rebus' beautiful image that he created for the New England Journal of Medicine Review. And the idea is that during the effector phase, when T cells come in and they try to kill the tumor cells, they release some interferon gamma. As Janice Taub and Suzanne have showed very nicely, interferon gamma, especially in melanoma, leads to upregulation of PD-L1. PD-L1 then sends a negative signal to the T cell, and the T cell can't effectively proliferate or mediate lysis. But what I'm going to show you a little bit is maybe it operates in this other phase, in this priming phase, because you know, and I just showed you, right, that PD-1 comes up very early in T cell activation in certain models, especially in tumor and self-antigen models. So does PD-1 have an effect in the initial stages when a T cell sees its antigen presented on a dendritic cell? So how can we look at this, actually? It's not so easy, but we've done it 2 or 3 ways, and I'll show you that the data come to a similar conclusion. So here's the experiment. So we take mice that express self-antigen, hemagglutinin all over the body. We've actually done this with tumor antigen as well, but it's cleaner with the self-antigen. And then you put in antibodies that block PD-1 family members. So you put in antibodies to either PD-1, PD-L1, or to B7-DC. So these antibodies are blocking the T cells that you're now going to put in. So you're blocking the PD-1 axis before T cells see their cognate antigen. This will be blocking during the priming phase. So again, the antibodies are in place when the T cells see their antigen widely throughout the mouse. And this is the mouse I showed you in the first picture, right? This is where you get a lot of PD-1 and a little bit of interferon gamma, okay? This is a model of self-antigen tolerance, the same way that tumor tolerance mostly works, actually. Now here's the trick, actually. So now you put in your targets. So your targets might or might not have PD-L1. So we have actually knockout mice that have no PD-L1. So if PD-L1 operates in the effector phase, then these cells ought to be able to kill the heck out of cells that lack PD-L1, right? Because there's absolutely none. So there's no PD-L1 on the targets in the effector phase. It ought to be really killable. We also did this with PD-L2. The readout is as before. It's putting in these targets that are CFSE labeled and either holding peptide or not. And the readout turns out to be quite clean, actually. So the bottom line is you put in these two cells. This one is loaded with peptide, and if the T cells can kill, they go away. Okay, very simple. It's an in vivo CTL assay, and you can read it out in terms of percentage lysis. So here's the answer. So here's a positive control. Here's the same cells going into a mouse being vaccinated with a lovely vaccine, a listeria-based vaccine, and they all kill, and they actually don't care at all whether the targets have PD-L1 or PD-L2 or not. So this is the positive control. Here's the negative control, the cells going in, being tolerized, and not killing. But check it out, actually. So if the targets lack PD-L1, doesn't matter. Doesn't matter at all, actually. So there's not really increase in killing by targets lacking PD-L1. This is targets lacking PD-L2. A little bit of increase there, but again, this does not really statistically significantly change killing. This is during the effector phase. What about if you block PD-1 initially? So if you block PD-1, you restore killing a little bit in no matter what the targets have. And you see a little bit, actually, but it's not statistically significant if the targets lack PD-L1. So the bottom line is in this model, it looks like PD-1 is operating more in the priming phase than in the effector phase. And in this model, actually, turns out that blocking PD-L1 is even more effective. So this is actually fascinating. This is a model of really profound tolerance. And if you block PD-L1, you completely restore effector function. These cells here are killing as well as a non-transgenic mouse with no cancer or no antigen hit with a vaccine, actually. So really, PD-L1 completely restores effector function. And again, the presence of the L1 on the target in this model is not as important as blocking during the priming phase. Now, this is a tricky experiment to interpret, right? Because I just showed you there's more cells, right? There's definitely more cells. So do you see more killing because there are more cells or because there are better cells, because the cells have a difference in effector function? That's a really hard experiment. The way to do that is to take these cells back out of the mouse and to do in vivo CTL analysis to see if you block PD-1 during initial antigen encounter, do you create cells, do you program cells differently? So on a cell-per-cell basis, they can kill better, okay? So Monica Goldberg in the lab did this, actually. And again, it's a challenging experiment, but the answer is actually quite clear. So here's the cells. This is the negative control. You take CD8 cells, you put them into a tolerizing environment. And again, this works both in tumor and self-antigen, but this is self-antigen because it's easier and cleaner to show. So here's cells that are tolerized. They don't kill. Listeria is your positive control. This is killing. And now we've blocked PD-1 during initial antigen encounter, take the cells out, and see if they now have a difference in effector function with no antigen around, actually. And it turns out blocking PD-1 completely restores, on the cell-per-cell basis, the ability of these cells to kill. So again, it suggests that that blocking has some effect during the priming phase. If you block PD-L1, it's even more surprising. Actually, the cells kill better than the virally vaccinated, the Listeria vaccine, actually. So this is blocking PD-L1 during initial antigen encounter. So again, two pieces of data suggesting in the same model that blocking PD-1 during the priming phase matter. Okay, this was me, right? This was just all my stuff. Has anybody else ever shown this? Yeah, actually, it turns out that our good friend and colleague used to be at Hopkins, and we sort of miss him, is Chen, showed the same exact thing in a completely different sort of model. So this is another way to tolerize T cells and to have them upregulate PD-1. So what you do is you take ova-specific T cells, you stick them in a mouse, and you give them ova-peptide with no adjuvant. With no adjuvant, PD-1 comes up, and the CD8 cells are beautifully tolerized, actually. You test them how tolerized they are by vaccinating them again with ova about 3 weeks later. And what you can see is without using any antibodies, the cells expand a little bit here, but they are completely anergic. They're refractory to restimulation, okay? If you block PD-1 early, right after you put these cells in, actually either at 0 hours using either anti-PD-1 or anti-PD-L1, you can restore their function, right? If you wait 24 hours, now the cells are seeing their antigen, right? And you block PD-1 there, you've restored a little bit. If you wait 48 hours, it is too late. So the bottom line is, once again, blocking PD-1 during initial antigen encounter seems to have a profound effect on their eventual fate. So what does this mean clinically for us? So one thing that it means is that anti-PD-1 monotherapy in patients probably is working, and this is not incompatible with the data on PD-L1, it's probably working on patients that might already have an active T cell response to tumor. And you can tell that because they have T cells in the tumor that are trying to make gamma, and the tumor is responding to that gamma in adaptive resistance by upregulating PD-L1, right? So again, this is certainly compatible with existing data. But what it means is going forward, actually, is if you want to combine PD-1 blockade with something else like adoptive T cell transfer or with vaccination, maybe you ought to think about having that blocking antibody there during the initial antigen encounter, actually. So giving it either before the vaccine or potentially even with radiation therapy to have PD-1 blockade in place as the radiation therapy proceeds. And again, there's actually another set of data using this in testing PD-1 blockade during initial antigen encounter versus trying to reverse exhaustion in a model of vaccination in macaques as well, using the Serrano antibody. So again, I think that these data are just, and this is not to say that you cannot potentially reverse exhaustion in some systems, but this is just to point out, I think that there are a fair amount of data that this operates during initial antigen encounter. Now, you know, Dr. Anthony spoke yesterday, actually, and he actually, I think, gave the folks who are looking at PD-1 and LAG-3 blockade a little bit more credit, because we actually were looking at LAG-3 on regulatory T cells for a long time. And we looked at it on CD8 cells, but we never really thought, honestly, of looking at PD-1 and LAG-3 together until we did this experiment. So this is an experiment where you take T cells and you put them in a mouse and you allow them to see self-antigen. They expand to self-antigen and they become PD-1 high. We're very, very fortunate that we're working with a vaccine. It's a very unique vaccine, an attenuated listeria vaccine that drives T cell division without upregulation of PD-1. It's a very reliable finding, and we published this a little bit, actually. But we wanted to understand what's the difference between the same cells in vivo that either upregulated PD-1 or did not upregulate PD-1. And to do this, we did a microarray. We adopted to transfer the cells. And Monica Goldberg, I believe, did a wonderful job on this. What she did was she took cells that only divided 3 to 6 times. So she controlled for differences in proliferation. And the starting population of cells in each animal exactly the same. It's from the same pot of cells. So these are genes that are expressed in T cells that are PD-1 high, that are non-functional. And of course, you see a nice positive control, which you don't always see in these experiments. And that is these cells here have PD-1 high. So that's actually good. In terms of genes that are expressed on the PD-1 not expressing cells or on the PD-1 low cells, you see effector genes like granzyme A and granzyme B, TBET, and granzyme K, so other things that are associated with better T cell function. So it seems to sort of make sense. So in this experiment, we saw, again, it was a little bit revealing for us. We saw, oh my gosh, there's LAG-3. So the T cells that have seen self-antigen and upregulated PD-1, at least on a bulk population, some of them had upregulated LAG-3, so these two markers. And this is around the same time, this is a little bit of older data, this is around time that John Werry was doing similar studies in LCMV models. We also have some other models, other molecules consistent with CD4 cells that have a regulatory function. So these data point out that perhaps some of these PD-1 high cells are not being deleted because they maybe have a regulatory function. There are a little bit of data to suggest that. These are norepinephrine, Norotin, and this is a molecule called Helios associated with CD4 regulatory function. You know, because we had so much luck, right, working with PD-1 knockout mice, we figured, look, the best way to do this is to breed the PD-1 knockout mice to LAG3 knockout mice, right? Then we'll have ideal cells, and we could study how these molecules interact. And we were actually doing this experiment in collaboration with Dario Vignali at St. Jude's, and the plan was actually really pretty good. So we were going to work on the CD4, on the CD8 cells, and he was going to work on the CD4 cells on an antibodies. So we were kept in touch, right? And we call him up like, hey, Dario, how are your mice doing? Oh, they're just fine. I'm like, Dario, you know, this is not working out. All our mice are dying, actually. And it turns out that what happened was his postdoc was mistyping them. And when they looked at their colony, it was the same thing. So in our colony, PD-1 mice do perfectly well. They have a normal lifespan. And we've actually done necropsies on the mice in our colony. And unlike some other knockout strains, we don't really see any evidence of autoimmunity. LAG3 single knockout mice are similar. They live a normal lifespan. And again, in our colony, we see no evidence of autoimmunity, even in older animals. But the double knockouts die actually usually early on, about 6 to 8 weeks of age. Not all of them die, actually. And what they die of, I actually forgot to put in the pathology slide, they develop very subtle signs of inflammation. This is not at all like a CTLA-4 knockout mice where they have widespread lymphoproliferative, what looks like a lymphoproliferative disorder. These mice have subtle signs of inflammation in the liver, in the heart, and really a tiny bit in the lung. What's also puzzling is people ask me all the time, you know, okay, what kills them? If you look at the inflammation in the mice, it really doesn't seem sufficient for them to die. What's really frustrating is you go down to get a set of mice to do an adoptive transfer experiment that one day before they look quite nice, and the next morning you go to harvest them, they're all dead. So one thing that we postulate is maybe the inflammation in the heart is involved in the conduction system, and they might have mouse studying cardiac death, and we don't have any mouse Holter monitors to assay that at this point. So the question, though, that those data has, the microarray data are interesting, right? They're nice, but they actually don't tell you whether the two molecules are expressed on the same cell. And Dr. Zerora did a nice job showing this, but I just want to highlight it as well. So these are CD8 cells and CD4 cells that infiltrate tumors in various mouse models. And the point is that PD-1 is expressed on the majority of cells, okay? The majority of tumor-infiltrating cells, both CD8 and CD4, express PD-1. Some of those cells express LAG3, not all of them. So there's so these are the double positive cells, LAG3, PD-1 double positive, and the CD4 compartment and the CD8 compartment. What you don't see, and I want to make this point, is what you don't see is LAG3 single positive cells in tumors. It's very, very rare, actually. And I think what Dr. Zerora showed yesterday in his really nice work, actually, is that you could have written TIM3 here, too, because you very rarely see TIM3 single positive cells. So it seems like if you look at the hierarchy of these immunological checkpoints, that maybe PD-1 is sort of the underpinning, that you'll express PD-1 and then you add something else on, like LAG3 or TIM3 or maybe BTLA to control T-cell function, but that the majority of cells do express PD-1. In terms of function of this, we did a series of blocking studies. These were done both at Hopkins and at St. Jude's, and actually in collaboration with Dr. Alan Corman of BMS, who used to be at Medarex. And these are some treatment studies, and this is with MC38. We see the same thing with B16, early stage B16, SA1N, and several other models. And so here's what you see. So if you treat these mice with no antibody, the tumors grow, and these are multiple tumors growing. In this model, MC38 is a colon tumor model. PD-1, this is one of the few mouse models that PD-1 is reliably functional. So if you block single block PD-1, you see some effect in tumor growth. And in fact, you can wind up with somewhere between 30 and 50% of the mice being tumor-free. LAG3, okay, LAG3 by itself does not cure any of these mice. Blocking LAG3 as a monotherapy, in none of the models we've seen results in any of the mice cure. What it does is it sometimes slows down tumor growth pretty substantially, and you can see here if you calculate the statistic called tumor growth inhibition, it is statistically significant. So it slows it down a little, but it's not curing anybody, any mouse body. On the other hand, if you combine the two antibodies, you can wind up with a significant fraction, here it's 80% of the mice, being cured. If you do mathematical studies for synergy, this is not additive. This is a synergistic effect, actually. So the bottom line is PD-1 and LAG3 operate through different mechanisms intracellularly, and probably they're upregulated differentially. And if you block them together, you can have effects that you don't have with blocking either one alone. And this is something we hope to try in the clinic soon. So bottom line, actually, and I did a little early because I know you have a lot of data, so Suzanne's great. She's let me here talk ahead of time. Okay, so how does PD-1 blockade work? The conventional wisdom by Dr. Rebus's editorial and generated in multiple systems is that it operates during the effector phase. Certainly the effects are clear. You have more cells, they're more functional on a cell-per-cell basis, and they traffic differentially, and those are data that I didn't show you. I showed you some data to help you think about it a little bit different way, and again, data from multiple labs and multiple models, that there is some effect of blocking PD-1 in the effector phase. And the reason I showed that, because as we think of combinatorial approaches with adoptive transfer with vaccines, I think that's something to keep in mind. I showed you that combining PD-1 with LAG3 is not additive, it's synergistic, and hopefully it will look like that in humans as well. In terms of the folks who did the work, Monica Goldberg was really a wonderful postdoc in the lab and a graduate student. She now left for Janssen, and she's apparently going to do great things there. Some of the data were generated by another graduate student, Chris Nerschel. The leader of our melanoma program is Suzanne Tabalian, who will be speaking next, and Julie Bramer has led some of the phase 1 clinical trials. Alan Cormett at BMS is a wonderful collaborator. We're thankful to work with him. Of course, my friend Dario, and funding from many organizations. This is the Melanoma Research Association, right? So now we have some funding from the Melanoma Research Association to really explore those really fascinating listeria-based vaccines that drive T-cell division without PD-1 upregulation. And I have like 1 second left or something, but if I had more time, I would show you more data of how that works. But I think this is a wonderful project, and these are really interesting vaccines. So I think with that, I think it's probably the end. And I don't know whether you do questions or I'll ask the chairman.